Uh, this is your practical paper today. You must read the entire paper before starting your experiment. Uh, Glucose is an important substrate for cellular respiration. Glucose is absorbed into the bloodstream from the small intestine and transported in blood plasma to the body cells. You are required to prepare different concentrations of glucose solution G. You must underline uh, what concentrations you have to prepare. Carry out the Benedict test. You should know the Benedict test. This is the food test for reducing sugars. On each of the concentration of glucose solution you have prepared. Carry out the Benedict test on solutions representing blood plasma P. So you have been given a solution labeled P, which is which represents blood plasma P. Use the results of the Benedict test to estimate the concentration of glucose in P. Actually, in this experiment, uh, and these are you are provided with the following solutions and materials. Label G 1% glucose solution, W distilled water, P unknown concentration of glucose, Benedict solution. These are the volumes given. What you have exactly to do in this experiment, you are given 1% glucose solution. You have to prepare concentrations of 1% glucose or dilutions of 1% glucose solution. How you will prepare dilutions? From 1% you can prepare, this is 1%. So you can further dilute it to half, which will be 0.5%, and you can further half make half of this, which will be 0.25, and you can further make half of 0.25, which makes around I guess 0.125. You can otherwise divide this with 2 and you will get this value, and you can also take half of this, whatsoever the value you get here, you take half of this. So at least you have to make 5 concentrations or 5 dilutions of this given solution: 1%, 0.5%, 0.25%, 0.125, and whatever the value you get here by dividing 0.125 by 2. So 0.125 divided by 2. So whatever the value you will get here, this would make this last percentage here. So you have to prepare these concentrations and carry out the food test with each of these concentrations. So when you will carry Benedict test, these may change some color, and or you may have to record the time taken for each time taken for the first appearance of green in each case. uh, and the same experiment has to be carried out with this blood plasma P sample given, and so the color of P produced, for example, let's see what you have to do. Yes, here you can see record the time taken for the first appearance of color change. If a color change occurs during this 60 seconds, so record the color after heating for 60 seconds. So I'll explain it later, but the point is you have to uh, heat, you have to add how you will perform this Benedict test for each sample. Take 2 cm cube from this 1% solution in a test tube, 2 cm cube, and then add 2 cm cube Benedict solution in it, Benedict solution, and then heat in a water bath at 85 degrees Celsius. Start uh, timer and record the time taken for the first appearance of the green color, or the first appearance of the color change in this test tube. So you will record time for this, you will record time for this, you will record time for this, you will record time for all these concentrations. You will also record time for P. For example, if this is 25 seconds this is maybe 40 seconds, this may be 60 seconds, this may be 70 seconds, and this may be, for example, 80 seconds. And for P, for example, you have 40 seconds. The, this also has 40 seconds color change in 40 seconds, and this has changed the color in 40 seconds. So it means that the concentration of blood plasma is compared with this, and the concentration will be 0.5% or whatever the value you get after doing this experiment. So this is basically your experiment. I'll repeat what is your experiment. You have to prepare the concentrations or dilutions of this, at least 5 dilutions made this way, and then you have to carry out Benedict test on each this concentration, and you have to carry out the Benedict test on P as well. How you have to prepare these concentrations or these dilutions and how you have to carry out the test, I'll explain that later. You're required to make a serial dilution. I'll explain later what is serial dilution and simple dilution of 1% glucose solution, which reduces the concentration by half between each successive dilution. So if this given concentration of the stock solution is 1%, you have to make it half. Half of 1% will be 0.5. So then again, half of this will be 0.25, and then half of this will be 0.125, and then you take half of it. So you are required to make a serial dilution of 1% glucose solution G, which reduces the concentration by half between each successive dilution. So this is half of this, this is half of this, these are the successive dilutions. You will need to prepare 10 cm cube. You have to prepare 10 cm cube. Figure 1.1 shows the first two beakers you will use to make your serial dilution. Complete figure 1.1 by drawing the extra beakers you need for your serial dilution. For each beaker, state under the beaker the concentration and volume of the glucose solution available for use in investigation. Available for use in investigation. Use one arrow with a label above the beaker to show the concentration and volume of glucose solution added to prepare the concentration. Use another arrow with a label above the beaker to show the volume of W added to prepare the concentration. So let's see following these instructions here. So let's see, two beakers have been drawn here. You should make at least 2 or 3 more beakers here, 2 or 3 like this. So if this is 10 cm cube available volume of 1% glucose solution, so you have to take half of this. So half of 1% will be 0.5, and you have to take 10 cm cube of this is available volume in this of 0.5% glucose solution to use. And how you can make 0.5%, you have to take half of it. So equal volume of this and distilled water should be added into it. So what equal volume should be added so that the total volume made here is 20, and then you will take 10 out of it, and the remaining will be 10 cm cube here. For instance, if you take 10 cm cube of this added into it and 10 cm cube of this would make total 20 cm cube here, but 10 has been taken out into the next beaker then, so available to use are 10 cm cube here. I'll write 10 cm cube instead of writing 20 cm cube here. Similarly, 10 cm cube from here, from this beaker into this, and 10 cm cube distilled water has been added into it. It will make total volume 20. I will not write 20 here. Why? Because I will take 10 cm cube from here and add up into the next, add it into the next beaker. So 10 cm cube. So remaining will be 10 cm cube of. I have to write concentration here. The concentration here will be half of this previous one. Okay, so if it is 0.5% here, so this will have half of this 0.5, so it will be 0.25% here. Glucose solution to use. Similarly, you will add 10 cm cube of 0.25% into it, and you will add 10 cm cube of distilled water into it. So this will have 10 cm cube of half of this 0.25. You know, this will have this concentration will be half of 0.25, so that would be 0.125 as we have discussed earlier. And so you make similarly you can take half of this value in the next beaker. So this is how you have to prepare these at least three more beakers, showing the arrows above and the labels below. Okay, so there are 3 marks. One mark will be for writing this correct labels here, 1 mark for having the correct volume added into it, and 1 mark for having the correct you know the number of uh, 3 more beakers added into it. So now proceed as follows. Set up water bath and heat to boiling, ready for step 6. This step, while you were reading it the first time, and you have gotten to know that you have to carry out a food test and water bath has to be used in this case, you should have set up water bath before even doing this step, or before even reading further. Okay, so that meanwhile you read this question paper, your the temperature of water bath that reaches to around 85 degrees Celsius. Okay, so prepare the concentrations of glucose solution as shown in figure 1.1. You should take against every point that you have completed. So after you have set the water bath, you should put take here, and you should put take here after you have prepared the concentrations of glucose, as we have by using the method as have been discussed in this diagram. Put 2 cm cube of 1% glucose solution into a test tube. Take a test tube and put 1 cm cube of glucose solution into it. State the smallest division on the syringe you used in step 3. So you look at the syringe and see what is the smallest division there. Maybe 0.2 cm cube, or maybe 0.1 cm cube, or maybe 1 cm cube. See whatever the smallest division is there. Your syringe may have ml written, which is actually equal to cm cube. Okay, so state the actual error when using this syringe. So actual error will be half of the smallest division on the syringe. So whatever the smallest division is there on your syringe, that would taking half of that would give us an actual error. So you will write you will calculate it, and you will write plus and minus with that value. So if it is, for example, the smallest division is 0.2, so half of the 0.2 will be 0.1. So the answer will be plus minus 0.1 cm cube. You may lose marks if you do not show your working. Calculate the percentage error when using this syringe. So percentage error can be calculated by, so this is the smallest error. So percentage error will be